It is my pleasure to introduce our speaker for this session, Dr. Lloyd Allen. Dr. Allen is Editor-in-Chief of the International Journal of Pharmaceutical Compounding, IJPC, and Remington, the Science and Practice of Pharmacy, CEO of the Midwest Institute of Research and Technology, and Professor Emeritus of the University of Oklahoma College of Pharmacy. He taught at the University of Oklahoma College of Pharmacy for 30 years, where he was a professor and chair with the Department of Medicinal Chemistry and Pharmaceutics. Dr. Allen's complete bio can be found on the Speakers tab in the left-hand menu. Please join me in welcoming Dr. Allen.
Good afternoon, and welcome to the session on quality compounding and clinical pharmaceutics. I've always enjoyed coming to the QCS meetings each year because there's some excellent speakers, and it gives me a chance to really interact with a lot of people. So I feel privileged to get to present again at this session. When we look at quality compounding, what we are really looking at is eliminating errors in compounding. And then when we get to the part of the presentation on clinical pharmaceutics, this is basically the scientific application of pharmaceutical principles to the clinical patient. So anyway, it's an interesting area, and we might as well go ahead and get started. And first of all, I don't have anything to disclose as far as conflicts of interest. You know the position I have with the Compounding Journal, and there's nothing in this presentation that it results from grants or anything like this. Now, concerning the learning objectives, at the conclusion of this program, each individual, whether pharmacist or technician, should be able to describe the different categories of pharmaceutical compounding errors. And in addition, list at least 4 common errors that may occur in compounding. You know, compounding is very detailed, very technical, and sometimes it gets somewhat complicated, and there's a lot of opportunities for errors. And so we're going to be looking at quite a few examples of those over the next half hour or so. And then another objective is to discuss common methods of avoiding compounding errors. And when we get into the area of clinical pharmaceutics at the end of the presentation, you should be able to list 3 pharmaceutic factors that can be involved in clinical pharmaceutics. So we're going to start with quality compounding, eliminating errors, with an introduction to errors, and then we'll look at a classification as well as several examples of errors. Then we'll look at how to avoid these errors. And then when we get to clinical pharmaceutics, we will have some definitions, and then look at all the many factors that are involved in clinical pharmaceutics, go through a number of examples on how pharmaceutics has helped to resolve different issues. Now, when we look at errors, what is an error? Basically, an error is an assertion or an act that unintentionally veers from what is true. In other words, it is an error that can end up in a preparation, a compounded preparation, that is not exactly the same as what it was intended to be. It's not identical to what the physician ordered, etc. And then when we look at a medication error, that is any preventable event that may cause or lead to inappropriate medication use or to patient harm. And so we're going to look at a number of these as we go throughout the presentation today. Now, there was a summary that has been presented by the National Coordinating Council for Medication Error Reporting and Prevention that occurred between 2001 and 2015. They have a large number of errors, and so they have categorized those and looked at what the result was and what the cause was, as well as how they can be eliminated. But anyway, some of the errors that are listed there include contamination. And obviously, when you look at contamination, we immediately think of sterile preparations not being sterile, but it can also be a non-sterile preparation that has been contaminated with bacteria, mold, yeast, or whatever. So anyway, contamination is one of the errors that has been reported. The compounded preparation may be super potent or have too much active pharmaceutical ingredient or API in it, or it may be subpotent and not have sufficient active ingredient. Some of the adverse events that result from errors can include infection, a systemic inflammatory response, and this one was reported from ophthalmic application, some corticosteroids, etc. So it's interesting to look at how these things were reported and what caused the error. Another adverse event would be an overdose. Blindness, even death can occur. You know, as far as compounding is concerned, you know, in pharmacy, we realize that errors are going to occur because of the complexity of compounding especially, but we have to work and do everything we can to minimize them. Another adverse event was diminished visual function. And what we're going to look at now is the different classifications of errors that the council came up with. We'll start off with some general errors, then look at errors involving incorrect ingredients, and then errors let me go back a slide here. Errors involving incorrect ingredients or incorrect concentrations, even the incorrect use of equipment, and we're going to spend a little bit of time on that. Then there'll be some physical chemical issues that are involved, like incompatibilities, and it's interesting that these also have a relationship to the clinical pharmaceutics that we'll be discussing at the end of the presentation. Errors can also involve microbiological contamination, even analytical testing, whenever samples are sent to the laboratories. And it's critical that samples be handled and prepared properly prior to sending them to the laboratory. And then there's some microbiological testing issues. This can be either in-house or at an outsourcing laboratory. And then there's a lot of miscellaneous errors that can occur. What are some causes of errors? Probably one of the primary causes of errors, especially for someone just starting into compounding, is the lack of education and training. You know, years ago, the pharmaceutical curriculum for pharmacy contained a lot of science, a lot of laboratory application and training, but whenever the degree was changed to the PharmD degree, there had to be room made for a lot of the other coursework to lead to the PharmD degree. And so it's sad that a lot of the scientific courses and laboratories were eliminated. And so we have been experiencing what the result was of the decrease in scientific education and laboratory training in the pharmacy curriculum. So lack of education and training is one of the causes of errors. Another would be the lack of implemented standard operating procedures, or SOPs. It's critical that each individual that is involved in compounding be thoroughly aware and knowledgeable of all of these standard operating procedures in the area in which they work. And they need to review these at least annually because this serves as a refresher, not only for them, but it serves as an opportunity to enhance or modify the standard operating procedures in order to prevent errors in the future. Another cause of errors is going to involve the improper use, maintenance, and calibration of equipment, improper chemicals and supplies, calculation errors. And incidentally, calculations is probably the primary cause of errors in compounding. And so we're going to talk a little bit about that later, about how to minimize errors in calculation. Another error is the improper form of the drug, whether it's the base form or the salt or the ester form or the hydrate form, or if it's a hydrate form, the proper hydrated form, because as you're aware, there are many different hydrates of different chemicals, and so it's important to use the correct one. And one thing I'll go ahead and add at this point is some pharmacists on their stock containers, when they receive them, if it is a salt or an ester or a hydrate, they may highlight that form with a marker so that it's evident whenever the compounder picks it up to use it in the compounding process. So very simple thing to do, just highlight whether it's a salt or an ester or which type of a hydrate that it may involve. There are other errors or causes of errors. It could be incorrect potency. And one of the problems we have too is the use of commercial products with unknown actual strength. You know, even though something says it's a 250 milligram capsule, or it's an oral liquid at 20 milligram per milliliter, or an injection at 10 milligram per mil, those are the labeled quantities. And as you're aware, the USP allows variation for these commercial products. The FDA approves the variation, and oftentimes it ranges between 90 to 110%, or it may be 95 to 105%. But most people are not aware that there's a lot of things that may be 80 to 120%. There's one or two items that may be not less than 90%, and one that may go up to 140%. So anyway, be aware of the allowable variation when using commercial products, because if you prepare one using a commercial product that's supposed to be 250 milligram, but it may range between 225 and 275 milligrams or so and still be approved, then whenever you make your product, it may actually fall outside the compounded allowable range of 90 to 110%. And we published several examples of that before, so just be aware. It would be nice if the manufacturers would actually provide a certificate of analysis with each of their manufactured products, but that just doesn't happen. Another cause of error would be lack of documentation, which can be quite an issue. I remember one time right after I graduated from pharmacy school, this is in the late 50s, I was working in a pharmacy and compounded a preparation for this patient, this lady, and she came in and I presented it to her and provided to her, etc. And she says, that doesn't look right. It doesn't look as strong as it usually is. So when the pharmacist came in later, I asked him about, you know, what the situation was, and she said it wasn't as strong as it usually is. So he looked at the formula and said, oh, well, it says here 3 drops of the red color amaranth solution, but I always put 10 to 12 in it because it makes it look stronger. And so he did not document what he actually prepared. And so that was an issue, obviously. So any variation in a formulation needs to be documented. There can also be misinterpretation of orders or formula instructions. So this is an example of where anything that is unclear needs to be clarified prior to proceeding. And then some people do not really focus and provide complete attention during the compounding process, and this can result in errors. Some of the general errors that came from the National Coordinating Council involve incomplete or incorrect information on the prescription itself, improper interpretation of the prescription, transcription errors, especially coming from a medication order in a hospital or a prescription in a community pharmacy, in transcribing it and putting it into the computer there is the opportunity for errors to occur. And then one of the initial steps in compounding is to do the necessary calculations. And we'll talk about that again in a few minutes, but incorrect calculations are a primary problem in the area of compounding. And another has to do with mismatched units and misinterpreting micrograms for milligrams, and all of a sudden you got a factor of 1,000 that you're off, and that can really be an issue. There can be some incorrect ingredient errors, such as an incorrect API. If you go back to your stock and pull the incorrect active ingredient, this would result in an error. Or you may pull the incorrect form, whether it's the salt form or an ester form of the drug, or whether or not it should be a pentahydrate or a sesquihydrate or a septahydrate. In other words, there's a lot of caution that needs to be observed in selecting the API and the proper API form. Not only the API, but the excipients. The proper excipients are critical because, as we all know, an excipient can really change the effectiveness of a preparation, whether it's compounded or manufactured. And then another would be the use of incorrect supplies, packaging supplies, etc., improper syringes or tubing or anything like that. So anyway, this could result in an error. And sad to say, there could be an error with an unintended presence of another medication, misreading of a label, or something like that. There's a lot of opportunities for error. Now, regarding incorrect concentration errors, obviously there's going to be either too much or too little. What we shoot for is just the right amount, but there's some errors that can be prevented. We'll talk a little bit about those here in just a few minutes. Another thing that might occur, especially with liquids, whether it's an emulsion or a suspension, would be stratification and the lack of a uniform appearance. You know, whenever a patient receives the medication and they look at it, if it's not uniform, you know, if it's separated and doesn't look right, they're going to question not only the effectiveness of the drug, but the effectiveness of the pharmacist that compounded it. So anyway, this needs to be addressed. There could be lack of uniformity of capsule weights. Of course, I think most compounders now that do capsules do check their capsule weights, and this is great. This is what should be done. There could be improperly filled or non-uniform suppositories or medication sticks, too much or too little powder, lack of proper formulation, lack of proper procedures, too much heat, too little heat. All of these can result in improperly filled or non-uniform suppositories. For example, if you're filling them and you've got your melt and you start cooling it down too quickly, and then you start pouring it into the molds and it starts solidifying too quickly and prevents the rest of the melt from flowing down to the tip, then it's going to be improperly filled. That could lead to an incorrect concentration of the drug or quantity of drug in the suppository. And believe it or not, when you look through a lot of these tables on errors that have occurred over the years, in some cases there's actually a missing drug. Now, one thing that was really shocking to me is a few years ago reading an article about compounding classes in colleges of pharmacy where the faculty would actually provide items for students to compound, and then they would analyze the preparations to see how close they actually were to the true quantity of drug that should be there. And the fact that there was actually some instances where there was no drug present was really quite concerning. Then we get into the incorrect use of equipment errors, not using the correct equipment. For example, and this is something that I would notice a lot in teaching in the laboratories, students would have their stirring rod in a graduated cylinder, stirring up a preparation, and then they would QS with the stirring rod still in the measuring cylinder. And obviously this is going to result in an error. And another similar thing is if you got a magnetic stirring bar in the beaker and you have a magnetic stir, and then you have the beaker that is calibrated, and so you then QS it while it is on the magnetic stir, but it has a stir bar in it, then obviously that is also going to result in an error. Another major error is a pH meter not being calibrated just prior to use, and incorrect storage of the pH probe is another problem. Another would be electronic balance not calibrated daily. I mean, after all, how long does it take to check your balance and to make sure it's calibrated properly? You know, it's not a procedure that takes an hour or two, you know, a matter of a few minutes, and make sure that you document it each time. Now, let's move into some physical chemical issue errors. Non-homogeneous mixing. Now, this can occur whether you're doing it manually or whether you're doing it using equipment. Used to, we just only were pretty much involved with mixing in mortars and pestles or beakers and things like that, but now we've got some incredible equipment that can be used to ensure thorough mixing. But that equipment must be used properly in order for your final preparation to be homogeneous like it should be. And another physical chemical issue occurs with solubility issues and precipitation. How many times have you initially dissolved an alcohol-soluble ingredient in alcohol, and then you added it to the aqueous vehicle, and all of a sudden it came out of solution and precipitated out? That was the incorrect order of mixing there. When you dissolve something in alcohol, and then you're going to dilute it into an aqueous vehicle, you've got to maintain the highest alcohol concentration at all times. And so this is done by adding the aqueous phase to the alcohol phase with mixing, not the other way around. This way, the alcohol phase retains the highest alcohol concentration while the aqueous phase is being added with thorough mixing. Another issue is failure to follow instructions. And then we can move into microbiological contamination of non-sterile and sterile preparations. We think of microbiological contamination mostly in working with sterile compounding, but microbiological contamination is also a big issue in non-sterile preparation, especially when water is present. Now, we've become very aware of water activity the last couple of years, especially with 795, 797, and beyond use dates. And so when you've got water present in a non-sterile preparation and it becomes contaminated, well, then that is a good opportunity for microbial growth to occur. And so non-sterile preparations can become contaminated, and that obviously should not occur. Now, we can have potential problems with laboratories, okay? And a lot of the problems occurs if the sampling is improperly done, if the sampling or the sample that's going to be sent is improperly handled, or it is not properly packaged or sent expeditiously. For example, it's best to send samples overnight. Never send them on a Friday where they would not get there until Monday. Don't send them the night before a holiday or anything like that, but send them overnight express so that the laboratory will receive it preferably the next morning. Another analytical testing issue has to be with improper beyond use dates. And this is something that we have been involved with for obviously quite a number of years. But all the beyond use dates today need to be assigned with stability indicating analytical methods. And so the easiest way of determining this is look at the references where the beyond use dates were published and make sure that they validated their analytical method, which is usually HPLC, by heating the sample to degradation, by oxidizing the sample, by adding acid to it and base to it to forcefully degrade the sample, and then modify the HPLC method in order to separate the degradation products from the intact drug. And so as long as that was done, then you should have a good beyond use date. And if it wasn't done, then that beyond use date obviously would be in question. Okay, we've talked a little bit about microbiological testing and some problems with laboratories. Let's now look at miscellaneous errors. What would happen to result in brittle suppositories of medication sticks? The primary issue here would be there's too much powder. You look at a 600 milligram boric acid suppository, or you look at some suppositories with zinc oxide in them or starch or things like that. The more powder you have, generally the more brittle they're going to become. And so generally, in order to correct that, if you take the powder and mix it with a liquid that is miscible with a suppository base. In other words, if it's cocoa butter, do a few drops of liquid petrolatum, and by mixing that with the powder and then incorporating that into the melted base, then that will tend to help keep them from getting so brittle. As far as if you're using medication sticks, let's say with a polyethylene glycol vehicle, and you've got two different molecular weights of polyethylene glycol, you can simply increase the percent of the lower molecular weight, the liquid polyethylene glycol, in the formula, and that should resolve that problem quite nicely. If you have oils floating on top of an oral or a topical liquid, this can generally be corrected by either adding a surfactant to it, especially if it's an aqueous product and you got an oil, then add an oil and water surfactant to it, polysorbate 80, tween 80, tween 20, something like that should work fine. And obviously, if it's an emulsion, then that emulsion may obviously already have an emulsifier or surfactant in it. And sometimes just by agitation in mixing, you can incorporate any additional oil that is added. If not, add the oil to a surfactant and then incorporate it. And that generally works out pretty nicely. Another issue that you see occasionally is when equipment is not properly cleaned. And so incorrect or insufficient cleaning of equipment, if there's any residue that is left on the equipment, that can result in carryover into the next compounded preparation, and that could result in some problems. Excess heating is a big issue because we tend to get in a hurry, especially when you start getting behind and have a lot of preparations of compounds, so you crank up the heat a little bit more on your hot plate. That's not a good idea because whenever you add your formulation to that, it's going to heat it up pretty quickly. It's a good idea to, on all your hot plates, to calibrate them. Know what the numbers on the dials, the approximate heat that refers to, and there's standard operating procedures to explain exactly how to do that. So if you know that you need to heat it up to 125 degrees, and you know that's going to be number 3 on your hot plate, then you can turn it up to number 3 and wait till it gets up there rather than just turning it up to number 5 and then hoping that, it's not going to get too hot. But calibrate your hot plate because it will save time in the long run. Now we get into some miscellaneous types of errors. And computer related is probably one of my favorite because one of the main issues with computer software in pharmacy is the software errors. Anytime you get a software upgrade, Well, first of all, it is important that it be upgraded, but sometimes these upgrades take a period of time to get accustomed to. And during this break-in period, errors can occur. It's a good idea to find out exactly what the upgrades involve so that when you're compounding using your new software, you can look for any potential problem and try to prevent it by being aware of it before it actually occurred. And computers not maintained is another issue. Because obviously a computer is one of your most important pieces of equipment in your compounding pharmacy or in your pharmacy period. And they've got to be kept in A1 shape, operating 100% all the time. How are we going to avoid errors in the future? Obviously, more and better education. Now, fortunately, a lot of the colleges of pharmacy are incorporating more and more science back into their curriculum and incorporating some laboratories back. Laboratories are very expensive, but it is important to better educate all the graduating pharmacists in formulation development and compounding. And there's a lot of schools now that have incorporated compounding courses in their curriculum, and this is great. And I wish all of them did it, and maybe they will at some point in the future. So to avoid errors, better education for pharmacists, more complete and better implementation of standard operating procedures, like I mentioned a while ago. Every individual needs to review the SOPs that they utilize every year, and if changes need to be made, suggest the changes to your pharmacy compounding pharmacist in charge and get any changes made that need to be made. Need to use, maintain, and calibrate equipment properly. A lot of the equipment needs to be maintained and calibrated by outsourcing it to appropriate companies. And this is really an excellent way to do it. For example, if you have them come annually, have them come at, let's say in May of each year, and that way you know when that they're coming. They can arrange the time with you, and you can make sure that your equipment is ready for them to maintain and calibrate it. Use correct chemicals and supplies. Checking each ingredient at least 3 times. We've all been taught, you know, check it, the chemical when you pull it from the shelf, check it when you weigh it, check it when you return it to the shelf, and make sure that the proper supplies have been selected for the preparation. And this includes everything from packaging to labeling, everything like that. Now, as I mentioned, calculations are the #1 area where errors occurred. All calculations must be checked by a second person, and potency calculations must be researched and checked by a second person. Now, this does not mean when you use a second person that you have written down the calculations on a sheet of paper and you give that sheet of paper to them and say, could you check this for me? Do not give them that sheet of paper. Give them the problem only, separately. Let that second person come up with the answer. And then they can compare, or you can compare their answer with yours. The issue with the first way of just giving them the paper to check it is there is not the incentive and the level of concentration that is required just by looking at something and seeing if it's okay or not. But forcing that individual to work through the problem individually and then check the answers is the best way of handling the calculation checking that needs to be done. Avoiding errors. Use a proper form of the drug. We've already mentioned that. Use commercial products of known strength, or use APIs. This is why it's always best to use APIs instead of commercial products if you can. In a lot of cases, you can't, especially in an IV admixture program. Sterile compounding may be a different situation where you have to use commercial products. But then we get into a different area that we'll mention in a few minutes, and that is the use of overfill. The USP allows so many milliliters of overfill depending upon a sterile vial, whatever its size is, and whether or not it's a small volume parenteral or a large volume parenteral. So we need to be aware of what those allowances are. And in some cases, it may be necessary to remove a part of the commercial product, if it's a vehicle or whatever, prior to adding anything to it. So anyway, this needs to be studied and followed appropriately. Determine the proper pH and use of appropriate buffers. This is especially important for stability issues. And interpret orders correctly. Don't guess. Focus and be attentive at all times. So there's a lot of information here in looking at looking at errors. And a lot of error prevention is simply common sense and paying attention to what is being done. And errors are going to occur, but it's our responsibility to minimize those errors, minimize the occurrence or the frequency of the error, and minimize the type of error that may occur. So anyway, honestly, when you get down to the bottom line, if you look at the hundreds of thousands of formulas that are compounded every day and those that are dispensed by pharmacists every day, the error rate is quite low, and that's something for all pharmacists to be proud of. We can always do better, but anyway, becoming aware of where an error may occur is a big step towards presenting it. Now let's go into clinical pharmaceutics for the next 15 or 20 minutes or so. Clinical pharmaceutics is the application of the unique knowledge base in pharmaceutics to patients and to clinical situations. Actually, compounding pharmacists have been involved in clinical pharmaceutics throughout history. We just really didn't know it. What are the factors involved? Okay, you remember one of the things you're supposed to be able to do is to list 3 or 4 factors. Well, here's a whole boatload of them. These are the factors that are involved in pharmaceutics and clinical pharmaceutics. Now, what's interesting, when you first look at this list here, what do you see? You see an entire course in pharmaceutics, okay? Here's a course that could take an entire semester to cover each and every one of these topics. But everything from sorption, whether it's adsorption or adhesion, buffer capacity, colligative properties, sodium chloride equivalents and all that, crystallinity, we're going to look at that here in just a few minutes. The dosage forms, the endotoxin levels for sterile preparations, hydrates, solvates. We mentioned a little bit about that earlier. Melting points, organic salts and esters, particle size, particle surface. And this is really important, especially for capsules and topical powders and things like that. Partition coefficients, where's the drug going to be in that preparation that you made, that emulsion or that transdermal gel that you prepared. pH is important, the pKa, dissociation coefficient, potency designations. A lot of things are not dosed necessarily by milligrams, but are dosed by activity. And this is where a lot of calculations can become involved and are very important. Then we look at precipitation and preservation, and rheology or viscosity and sedimentation for suspension. But rheology is also important in your topicals, your gels that you prepare. Solubility, very critical, as well as surface tension. Now, years ago, we've been involved in incompatibilities for many, many years. And this is a part of clinical pharmaceutics. I mentioned we've been doing clinical pharmaceutics for years. Well, incompatibilities is a part of that. When you look at incompatibilities, there are basically 3 types. There's a chemical incompatibility, there's physical incompatibility, and there's therapeutic incompatibilities. Now, when you look at physical incompatibilities, these can involve incomplete solution. You don't have enough solvent to dissolve the solute that is present. This can result in precipitation over time. If you prepare it in a room temperature and put it in a refrigerator, the solubility decreases, and so it's going to come out of solution. So this is a physical incompatibility that occurs. If you try to mix oils and water, obviously you've got two immiscible liquids. And so that's an incompatibility, but we can use it to our advantage by using emulsifying agents and preparing an emulsion. And then liquefaction of solid ingredients. This is where eutectics and deliquescent solids come in. And we can use eutectics especially to our advantage whenever you're mixing two liquids that are going into a topical preparation. Form the eutectic first and then incorporate it into the vehicle. It makes it a lot faster and a lot easier to come up with a uniform preparation. Or another physical incompatibility is the incorrect form prescribed, and we've already talked about that under errors. About chemical incompatibilities, what are we talking about? Oxidation, reduction, racemization, precipitation can be both physical and chemical. Gas evolution, especially carbon dioxide evolution, or color changes. An explosion obviously is a chemical incompatibility. You certainly would not want to try to compound that mixture. Cementation or hardening of a product. This occurs oftentimes with suspensions and things like that. Separation of liquids, especially occurring with emulsions. Gelatinization can occur. The evolution of heat and cold, you know what happens when you add potassium iodide to water and things like that. Hydrolysis, the presence of water in a preparation can result in hydrolysis. This is one of the primary stability issues that we have, especially in oral pediatric liquids, and that's why the on use dates are relatively short in many cases. Or polymerization, and this can actually cause gelatinization, but polymerization can occur with proteins and other drugs as well as excipients. Double decomposition, probably remember back from your Chem 1 days, where two chemicals come together and each can decompose, etc. Development of a poisonous substance, not so much of an issue today as it used to be. And then there's many other reactions that can occur. How can you correct incompatibilities? You got something that you know is not supposed to work. Well, the first law that you need to know is that they are easier to prevent than to correct, okay? Incompatibilities are easier to prevent than correct. You can modify a mixing order. We're going to see an example of that here in just a few minutes. Modify the mixing order. Or you can add an ingredient such as a suspending agent, emulsifying agent. You know, if something is not going to go in solution and you're going to have a suspension, there's nothing wrong with that, because actually suspensions in many cases have better taste properties and better stability properties. So why not just simply add a suspending agent and dispense or compound a suspension instead of trying to make a solution out of it? You can add a solubilizing agent or a diluent, stiffening agent or softening agent. Many different options are available. In some cases, you need to remove an ingredient. You can separate it into two prescriptions. Obviously, in a situation like this, it's going to be best to explain to the physician, because otherwise, when the patient goes back and they say, well, you know, those two prescriptions you gave me last time I was here, he says, I didn't give you two, I only gave you one. Well, in order to make sure there's no misunderstanding, check with the physician and you give him the option that it can be done into two separate prescriptions. Change the vehicle, ingredient, or dosage form, using labels, shake well, refrigerate, whatever is necessary in order to come up with a reasonable preparation. Now, there are some issues with excipients that you need to be aware of. Sulfites, we're all sensitive to those because they happen quite frequently. quite frequently. Benzalkonium chloride can cause paradoxical bronchoconstriction, can be a problem. Aspartame can cause headaches. Saccharin can cause some dermatological reactions. Benzyl alcohol can cause problems in kids and dyes, especially with sensitive patients. Propylene glycol can cause localized contact dermatitis. Now, when we look at specifically at pediatrics, patients less than 6 months of age, it's a real problem with benzyl alcohol, propylene glycol with topical dermatitis, etc., and sensitivities to polysorbates 20 and 80, and limiting the ethanol content in some cases may be advisable, and sensitivities to some preservatives can be an issue. And then there are patients with metabolic disorders, so need to be aware of the excipients that you are using. Not only that, but aware if you're using commercial products, be aware of the excipients in the commercial product. And we'll see how that impacts some of the products here in just a few minutes. Okay. Hypersensitive patients, especially to dyes, parabens, sulfites, and even lanolin for topical use. Now let's look at an example where particle size has been important. This is from the manufacturer's standpoint. What did they do that involved pharmaceutics? Well, years ago when griseofulvin first came out, it came out in 250, 500 milligram tablets. A dose was 500 milligrams to a gram daily. And this was in their micro-sized particle product. The thing with griseofulvin is it's not very soluble at all. It's very poorly soluble. And so their product was micro-sized at less than 4 microns. They made a new product. It was ultra micro-sized with less than 1 micron in particle size. But look at what's happened to the dose. The dose was decreased from 500 to 375 milligrams, or from 1 gram per day to 750 milligram per day. So a decrease in particle size resulted in an increase in the bioavailability of the drug. So this is the situation where particle size reduction is good. There's also a situation where particle size increase is good. The product nitrofurantoin, we used to have a lot of problem with it because it was very rapidly dissolved and absorbed, and it would cause nausea and vomiting. So what did the company do? They made a macrocrystalline form of nitrofurantoin called macrodantin. This would slow down the dissolution and slow down the absorption rate of the drug. And this worked out very good. However, they didn't stop here. They came out with another one, which had 2 particle sizes of the nitrofurantoin and a gel. The macro size slowed the dissolution rate, and the gel excipients in the tablets formed a gel or the capsule and slowed down the dissolution rate of the powder. So here's 2 pharmaceutical principles that were used in order to minimize the nausea and vomiting that occurred from the very rapid dissolution and absorption of the drug. There can be some bioavailability differences in commercial products that you dispense. Posaconazole oral forms cannot be substituted. The delayed release, believe it or not, has a high bioavailability. The suspension has a lower bioavailability. This is why it's important to look at the excipients in commercial products, because this delayed release form actually has dissolution enhancing ingredients in it. And so it can actually, even though it's a delayed release, the delayed release is separate from the excipients that cause the enhanced dissolution once the drug is released. So it's kind of interesting issue there. Valsartan suspension has 60% higher bioavailability than the tablets. Again, it's important to look at the excipients and see why this might occur. And so you may need to make dosage adjustments when changing from one form to the other. There was a situation of Phenytoin intoxication in Australia, and I've used this example before in case you've heard it, there was an outbreak of Phenytoin intoxication. And so the drug company, this was a commercial product now, not compounded, but a commercial product. Analysis showed that the drug content in all the capsules was fine, not a problem. But the manufacturer had changed the diluent from calcium sulfate to lactose. Well, what's the big deal? Calcium sulfate is only slightly soluble in water. Lactose is freely soluble. What is the first step in dissolution of a drug? Is wetting. The particles have to be wetted. Calcium sulfate did not take up the water to surround the phenytoin particles with, and so it was slow to dissolve, whereas lactose is freely water soluble, so it sucked up the water and surrounded the phenytoin particles, and they would dissolve faster. So this is a situation of a commercial product using pharmaceutics to change the issue that they had with phenytoin intoxication. So, this also can occur in compounded preparations too. There's some salt ester form issue we talked about a little bit under some errors. Erythromycin has a low solubility of about 1 milligram per mil. This is not a whole lot, but it is enough to cause a very bitter taste of plain erythromycin. So if you have a prescription to compound with just plain erythromycin, you don't want to use the erythromycin. But you look at the different ester forms, salt ester forms of the drug. We've got erythromycin estolate, which is practically insoluble. And keep in mind, an insoluble drug forming a suspension is practically tasteless, okay? We also have erythromycin ethyl succinate, very slightly soluble, which is just a little bit more than practically insoluble. So it's not very soluble at all, and it too is practically tasteless. So in this situation, we want to use one of these two forms, not the erythromycin, in order to get a tasteless preparation. But there's a difference. 250 milligrams of the erythromycin is equivalent to 400 milligrams of these other two forms, and so some adjustment will need to be made there. Another issue is order of mixing. This is what I referred to a while ago, which is very interesting. A formula was given for the pharmacist compound using Diazepam Injection, 20 milliliters, ethanol 95%, 5 mils, and propylene glycol 30 mils. And he was to mix it. This is going to be an oral liquid. He's going to mix that with methyl cellulose 0.5% solution mixed 1-to-1 with simple syrup. Okay, so what's the big deal? Well, let's look at it and see. What he basically did, first of all, was, oops, went one too many, sorry about that. The first thing that the pharmacist did in preparing this one was to mix the, come on, let me get back here. Here we go. Okay, was to mix the, I lost it again, I'm sorry. Okay, in other words, what he did was he took the methyl cellulose suspension and the simple syrup, mixed it 1-to-1 for his vehicle, and to this he added the diazepam, the injection, and then he had the propylene glycol and the methyl cellulose simple syrup, and a cloudy product was obtained. He could not dispense that. Well, this is where it's important to know what you're working with. Diazepam injection consists of 40% propylene glycol because diazepam is not water soluble. So it's 40% propylene glycol and 10% ethanol. Now, looking at this, the vehicle has got propylene glycol added to it. So this is going to be soluble in the propylene glycol and the alcohol. So let's do this first. Let's put the diazepam with the alcohol and propylene glycol, and then add that solution to the methyl cellulose and the simple syrup, and this will come out in an okay preparation. Okay. Another issue, captopril is not very stable in aqueous solutions, degraded by oxidation. If there's any metal ions present, and there is in a lot of the vehicles, and so simply add EDTA in order to correct that issue. Now, we're using a lot of oil-soluble drugs today. There's nothing wrong with using the fact that they are oil-soluble to compound a good preparation. They're soluble generally in triglycerides, soybean oil, things like that. Now, commercial products today include SEDS, self-emulsifying drug delivery systems. Now, these are commercial products, and they are microemulsions. They are pre-concentrates. Sandamune is one, cyclosporin, and what they consist of is the drug, vegetable oil, ethanol, and maybe a polyglycolized glyceride. But they form a microemulsion, and when that is taken orally, the aqueous contents of the stomach mixed with it form an emulsion, automatically forms an emulsion, which is quite bioavailable, and it helps to protect the drug, etc. And Cremophor RH40 is widely used, as well as Labrafil, and these are often used with propylene glycol as the co-solvent. With transdermals, in order to move right along, transdermals are used a lot to minimize GI distress. NSAIDs and PLO gels are especially true, used a lot. Now, there's a situation where androgel was prescribed for a patient, compounded, and the patient was also using a commercial miconazole product. He actually applied the androgel to the same area that the miconazole had been applied, even though they were applied separately. And what happens is he started getting testosterone toxicity. And the reason is that miconazole has some penetrating enhancers in it, and they remained on the skin, even after the period of time, had elapsed before he applied the androgel. And so these things do need to be applied to separate areas on the skin. And then comes the area of occlusion, whether or not to use an occlusive dressing. Enhanced hydration with transdermals can result in an up to 100-fold increase in drug absorption. So this should be used with caution, and make sure that the patient is well aware of the potential increase in drug concentration that they're going to receive. The driving force of penetration is thermodynamic activity of the API in the vehicle. Now let's look at this real quickly. A saturated dosage form at 25 degrees when you compound it at room temperature may not be saturated when it's applied to the skin at 32 degrees. So you prepare a saturated solution, and it is the concentration that is forcing the drug through the skin. But whenever you apply it to the skin at a higher temperature, the drug is more soluble, and so it is not a saturated solution anymore. So that can be an issue. One of the ways that you can counter this is to use solvent evaporation to increase saturation of the drug after it's applied to the skin. In other words, add some alcohol to the vehicle. As it's applied, then the alcohol evaporates, and it increases the concentration of the drug in the product that is applied to the skin. So last thing on Avastin real quickly, because we're running out of time, is silicone oil has been an issue in syringes and cartridges, vials, stoppers, etc. There's generally about 0.6 milligrams to 1 milligram of silicone oil per 1 milliliter per syringe, okay? So this gives you an idea of about how much silicone is actually used in the syringe or a vial stopper or whatever. Now, syringes are used off-label a lot in ophthalmology as common practice. And floaters occur on injection into the eye, and sometimes vitrectomy is required. But a study was published that's interesting that showed that if you flick the syringe when you're obtaining the Avastin and stuff, this causes increased number of droplets to be released. So the caution here is don't flick the vial or the needle. Withdraw the solution from a vial or ampule using a sterile filter needle and replace the needle prior to injection, okay? So don't flick and then use a filter needle, replace the needle prior to injection. And obviously don't agitate or don't flick. One last thing on paclitaxel infusion contains cremophor or polyoxyl 35 castor oil and ethanol. And what this can do is to actually extract part of the plasticizer, DEHP, diethyl phthalate, from standard infusion sets. And so this is why a non-PVC container and non-PVC infusion set is recommended for something like that. Real quickly, wrong suppository base results in seizures. Water solubility of a base can strongly influence the absorption of lipophilic drugs. The teen was prescribed with epilepsy was prescribed Diazepam 10 milligram suppositories. They were compounded using cocoa butter. The teen went to the emergency room twice because of seizures. What happened here? Diazepam is soluble in cocoa butter, and the escaping tendency, the drive to push diazepam out of the cocoa butter is not there. And so this resulted in a low absorption rate. It was corrected by using a water-soluble suppository base, because once the water-soluble base gets in the rectum, the aqueous mucosal fluids dissolve the vehicle. The diazepam will move into the water vehicle and then be ready for absorption. Okay, and there's some other things, but we're running out of time. But Concerta and some generics are not necessarily equivalent. The reason for this, even though they are FDA approved, they use different technologies to release the drug. Concerta uses a laser cut hole, that should be HOLE, and an osmotic pump method. Generics are not required to use the same method, so be aware of that. Special needs patients, there's a lot of those. They're intolerant to a lot of different things. And so in summary, I'm sorry to be a few minutes over, errors will happen but must be minimized. One error can result in years of litigation, literally years of litigation. So errors are a lot easier to prevent than to litigate after they occur. Pharmaceutics is unique to pharmacy and can be the difference in success or failure in therapeutics. Thank you. If you ever need any more information on any of these topics, just never hesitate to contact me at this email address here. And for credit, here is the claim code. And I really thank you for your attention for this presentation. Thanks a lot.
Hello, everyone. I hope you caught that CE code that Dr. Allen very kindly put up for you. If not, we'll say it a bit again after our live Q&A. So it is with my great pleasure to welcome him live and in person to answer your questions, the wonderful Dr. Allen. Dr. Allen, join us. Hello and welcome. Did you want to make any sort of summary remarks on that presentation before we move to Q&A?
Just a couple of things. Quality compounding involves basically error-free compounding. And one of the most interesting aspects of compounding, I think, and in fact, pharmacy practice, is the area of clinical pharmaceutics, because that involves the use of your scientific background directly applied to patient care.
That's exactly, yeah, I think it's been very interesting in these with watching this presentation and the things we've learned. There's a couple of questions that have come up that we wanted to see how you would respond to. Does the current pharmacy curriculum accurately and completely train a pharmacist for compounding? And if not, what should be changed?
Well, that's really a hard question to answer because pharmacy is very complex and consists of a lot of different types of practice. I remember back when we were changing from the basic curriculum to the PharmD curriculum, that there were a lot of science courses that were removed. And of course, when we start looking at clinical pharmaceutics, that's based on a lot of the sciences and the incorporation of the sciences into patient care. So I think that's one area that the curriculum could be strengthened. Another has to do with calculations because as we saw in the first part of the presentation, calculations are one of the biggest areas where errors are found. And so calculations are very critical. And one last area of involvement I think that needs to be enhanced has to do with the use of excipients. Pharmacy students learn a lot about the APIs, the active pharmaceutical ingredients, and they're quite well trained in that. But when it comes to excipients and how they can impact the drug effect and either make it or break it, I think there's more effort needs to be turned and more emphasis needs to be placed on the roles of excipients in pharmaceuticals. And thank you for the question.
You are very welcome. I'm keeping an eye in the chat for any other questions that might have come up. Maybe you can expand a little bit on what you were saying in your presentation about clinical pharmaceutics intervention. You spoke of that in your slides. Can you maybe tell us a bit more about that?
Sure. You know, with clinical pharmacy and the use of pharmacists more in therapy today, as we saw in the area of clinical pharmaceutics in the presentation, there's a lot of questions and therapeutic failures that can be explained by pharmaceutics. And so, you know, going into pharmaceutics and into a little deeper level, we can understand better why drugs may or may not be effective. And many pharmacists do practice in the area of clinical pharmaceutics, which is beyond clinical pharmacy, but there's a big need for even more to become involved because generally they begin with a commercial product that has been manufactured, but then we can solve a lot of the issues that may arise, as you saw in the examples, by pharmaceutical compounding and by the proper selection of the right drug, the form of the drug, the excipients. Then you can, using clinical pharmaceutics, identify the problem and resolve the problem to the benefit of the patient.
That's the ultimate goal, the benefit of the patient, right?
Right.
I
That's
noticed
true.
you've spoken a bit about your work both not only nationally but internationally on some projects you have regarding new drug product formulation. Can you tell us a bit more about that? And we've only got a couple more minutes and then we'll wrap up. So any questions, make sure you get them in the chat for Dr. Allen.
Oh, formulation is basically very similar regardless of what country you're in. And obviously, we're well aware of the fact that so many of our drug products are developed and manufactured overseas and imported into the United States. When it gets into compounding, it's a little bit different story because in some countries, the government tightly controls what can and cannot be compounded. And so you're kind of limited in what you can do and the variations that you can make in some countries. But fortunately, we have great communications worldwide among pharmacists, and by these communications, then we can help bring each other up to a higher level in quality compounding. And so obviously that's the goal for all compounding pharmacists throughout the world.
I think that's amazing, and any impact we can make around the world to, help on this topic is amazing. So thank you, Dr. Allen. I really appreciate your participation. You are definitely one of the original and kingpins in this topic. So we really appreciate you taking the time and your presentation today. Thank you again.
Thank you very much, and thank to all that attended. And if they have any questions, my email was on the last slide, so just let me know.